Hello everyone, this is Mr. Jackson, and today we're going to be getting into part 1 of our Module 4 lecture over the circulatory system, specifically looking at the blood vessels. Now, whenever we're referring to blood vessels, this is a closed vascular system, a one-way closed loop of a system that transports blood to the tissues and back to the heart. Remember, this is the one-way closed system that will basically transport blood to both the pulmonary and systemic circuits and to and from the heart. So, vessels that carry blood away from the heart, we're going to get a little bit more specific now. We're not just referring to arteries, we're referring to arterioles. And arterioles are, you can think of those as just the smaller divisions of arteries. So as we get closer to those target tissues, we're going to have a narrowing of those vessels. And so arteries will then become the more narrow, small arterioles. You have vessels that play a role in the exchange, the actual exchange between tissues and the bloodstream, which are your capillaries, your capillary beds. And then you finally have the vessels that will return blood toward the heart, which will be your venules and your veins. So same as before with arteries. So we've heard veins and arteries. The smaller, and you can think of it, narrower form of the returning vessels are called venules. And there are 3 layers, or what we call tunics, in blood vessels. 3 layers that apply to your veins and venules, your arteries and arterioles, and this does not apply to your capillaries. We'll go over why that is the case here in just a moment. But we've got 3 layers, and we'll cover them real quick from inner to outer. The tunica intima is the inner layer, or inner tunic, of your blood vessels, and this is really important because it's going to create a friction-reducing lining. So as blood and nutrients circulate through the blood vessels, we don't want there to be a whole lot of resistance that they have to contend with if we can avoid it. And so reducing any form of friction that the transported materials might come into contact with will be very, very important. So the tunica intima is all about reducing friction. The tunica media is made up of smooth muscle and elastic tissue, and does have a sympathetic nervous system controlling it. And so this is the really active layer. Whenever we think of any sort of vasoconstriction or vasodilation, any sort of actual like pumping of the blood throughout the vascular system, this is the layer that we're often referring to. So that tunica media being the active tissue. And then the final one, the most outer layer, is the tunica externa. And this is just good for forming a protective outermost covering. Okay, so it's mostly connective tissue, helps support and protect that vessel and anchor it to surrounding tissues. Gave you a visual here so that we could see some of this microanatomy of these blood vessels. And I do want to point out here, so we've got our tunica intima. They have that broken down. You can see that that is here on the innermost layers. We've got our tunica media, so just superficial to that intima. You're going to see that second layer here, which is the media. You can see that that's comprised of smooth muscle tissue, and it's a little bit thicker. Okay, so again, this is the more active layer of tissue. And then the tunica externa is that outer protective covering. So it does a really good job of showing those tunics for your blood vessels. Also does a really good job of showing the difference between an artery and an arteriole, right? So you can see arteries are larger than arterioles, and veins are larger than venules. And you can also see that if I were tracing the pathway of a blood cell through the circuits, I'm going to begin by carrying blood away from the heart through an artery. It will then enter an arteriole, and in order for it to arrive at the body tissues, it's going to eventually get to this capillary bed. Once we've exchanged the nutrient for the waste, or for whatever else is going to be gotten rid of after we have delivered it to body tissues, then at that point it will go from the capillary bed back into a venule, smaller vein, and then it will go to a larger vein back to that right atrium of the heart through either the superior or inferior vena cava. So there are some structural differences that we want to make sure that we understand between arteries, veins, capillaries. So arteries typically have a heavier, stronger, stretchier tunica media than veins do, because they have to be able to withstand those large changes, because they're carrying blood away from the heart, right? And so that means that they're contending with the pressure exerted from every single pump or beat of the heart. So it's going to be a lot heavier, a lot stronger, and stretchier to withstand that relatively higher amount of pressure that it's going to contend with. Veins, typically, they have a thinner tunica media because they're almost always under low pressure. They also tend to have these valves that help prevent backflow. Since there's less pressure and sometimes they have to go against gravity to return deoxygenated blood and waste products to the heart, we want to make sure that everything goes in a single direction. And if we're going against gravity, then those valves are going to be really, really important. The lumen of the veins, just that inner tissue portion, that inner space where the blood actually travels, that is going to be larger than that of arteries. Again, we want to make sure that we are able to deliver everything that we need to, and we can compensate from that lower, I would say, tunica media quantity. And then we also have this mechanism that's really cool, where we have like a skeletal muscle pump that essentially milks or helps manually deliver blood in veins up against gravity towards the heart. So if you think about like what allows the low pressure blood supply to actually still go against gravity and to reach the heart so we can return it to that right atrium, it's going to be the one-way system helping or helped out by valves, and then also the skeletal muscle pump. So anytime your skeletal muscles contract in a certain way, it'll actually create just sort of this milking effect where it takes it all in a single direction. Some more structural differences. So let's finish with the capillaries here. Now the capillaries, we said that arteries, arterioles, veins, venules, those have those three layers, right? The intima, media, and externa. But when it comes to capillaries, remember, this is the area where we're actually exchanging nutrients, right? Between the blood supply and the body tissues. And so we don't want those nutrients to have to pass through a whole lot of layers, right? We want to make sure that that's as easy of an exchange as possible. And so these capillaries only have one cell layer, and that is exclusively that thin tunica intima. And so this is going to create rapid exchange between blood and tissue. We do have networks that basically serve all the way throughout these body tissues. We call those capillary beds. And basically, we have our own little term that refers to the way that blood flows through these capillary beds, and we call it microcirculation. So it's basically going to go from one of those end or terminal arterioles that's carrying blood away from the heart, and then we're going to reach those exchange vessels in the capillary bed, and then we'll get to basically the other side of that. So now we've done nutrient exchange, and we've taken up waste products and deoxygenated blood and what have you, and we're now going to be in a post or after the capillary venule, which will then lead to veins that then lead back to the heart. So we do have one other thing that we want to make sure that we understand. It's pretty cool whenever it comes to physiology as well. But whenever we're looking at capillary beds, we do have two very, very important structures that we want to understand exist. And these are the precapillary sphincters and vascular shunts. Now, these precapillary sphincters are really, really cool for regulating blood flow through a capillary bed. When those are activated, they will close off certain capillary beds and redirect oxygen-rich blood. And these vascular shunts are direct connections straight through, kind of like superhighways between the arterioles and venules to opposite sides of those beds. So they're kind of like bypasses. So even if the capillary bed happens to be blocked off for some reason, which we can go into some examples here in a moment, the vascular shunt is going to make sure that we still have that, one-way flow of blood that's ongoing. We just won't be delivering as much to the actual tissues surrounding those beds. So again, when those sphincters are open, blood flows through and exchanges with cells of those tissues, but when we close them, blood flows directly through the shunt and bypasses those regions of tissue altogether. And I will say that this, I think, doesn't do the best job of showing the difference between these sphincters because they're not even here, and that superhighway that we referred to, that shunt, that would essentially go through the middle. But if you want to think of it here, the terminal arteriole, if you want to imagine a straight line highway from arteriole to venule, that is what we refer to as the shunt, right? So it's a one-way directional system. All of this extra to the top and to the bottom that you see here, that would be referring to the capillary beds that actually exchange nutrients with the tissues. And so those sphincters are essentially going to be at the beginnings of these arterioles. And if those close for a variety of reasons, then it will basically shut off blood supply to those tissues and just basically redirect it so that it can only go straight from arteriole to venule without exchanging as much in the tissues themselves, right? So where you have this full capillary bed that is currently has their sphincters open, and so it's delivering and exchanging all those nutrients. If we were to close down those sphincters, then you would expect for there to not be as much blood flow. Only thing I don't like about this image is that it doesn't really do justice for the fact that we're still going to be transporting blood through that shunt that connects these two. So we're going to get into some gross anatomy, and then we're going to conclude part 1. Now, we have major arteries of the systemic circulation. We want to make sure that we understand some of the key structures within that, right? And so our major artery of systemic circulation is going to be the aorta. It's the largest artery in the body, happens to leave from the left ventricle of the heart. And if you're looking at the aorta, there are several regions that basically gives rise to all of the smaller arteries that will actually take blood to specific tissues and specific regions of your body. We have the ascending aorta. That's the part that directly leaves the left ventricle. We have the aortic arch. So that's going to arch to the left. And that gives rise to a bunch of additional arteries. You have the thoracic aorta, which basically travels down through your thorax. And then you finally have the abdominal aorta, which is going to go through the diaphragm down into the abdominopelvic cavity. At this point, we're giving rise to arteries that are going to serve your lower extremities. And here's the deal. We are not going to, for the scope of this lecture, going to just basically go through and talk about the names of every specific artery and the specific regions they serve. But what we are going to do is we are going to want to focus in on right here. So if we have, for example, the ascending artery, we are going to break down through an activity in class which arteries basically stem from each of these portions of the aorta and what regions those serve more generally, right? So is it going to the head and neck? Is it going to the upper extremities, to the thorax and to the abdomen? Is it going to the lower extremities? We need to understand that this major artery, the aorta, is giving rise to all of the arteries at different branching points. We have two major veins of systemic circulation. This is the superior and inferior vena cava, which enter the right atrium of the heart. They return that deoxygenated blood so that we can then put it back into pulmonary circulation. The superior vena cava is draining the head and the arms, right? So that makes sense. It's coming from the top. And then the inferior vena cava is draining the lower body. Again, makes sense because it's coming up from the bottom. And just like before, so we had a full breakdown of the arteries, and now we're looking at the major veins. And again, this is really, you're wanting to look at the superior and inferior vena cavae. And you can see that basically we have a full breakdown of veins that drain certain regions, right? So we're not going to get too specific for the time being, but will in activities that follow. So the veins of the head and trunk are drained by these structures, these veins specifically. The upper limb, the lower limb. So we'll be able to break those down, but essentially what we need to understand is just superior, inferior vena cavae, and the fact that basically superior is going to be the upper body and inferior is going to be the lower body. And we're going to finish this first part over anatomy of the vascular or the circulatory system with one type of circulation that basically doesn't follow exactly this rule, and that's our hepatic portal circulation. So this is going to be formed by the veins that drain your digestive organs, your spleen, your pancreas, and empty into what we call the hepatic portal vein. And this is then going to carry blood from those locations to the liver, where we will process these nutrients or whatever products have been circulated or filtered into the bloodstream from those tissues. So from the digestive tissues, for example, we're going to carry that to the liver first, so that that liver can actually process a lot of those nutrients, and then we will then return the nutrients that aren't kept by the liver, essentially, and we'll return those to systemic circulation. And it's going to receive blood from these specific veins, but again, for the time being, we're just going to want to understand that it comes from these organs and systems. And we'll finish by giving you a really quick visual image of what this hepatic portal system would look like. Again, you have arterial blood being delivered to the, let's say, digestive system, like the stomach and the intestines. In those capillary beds, we'll absorb nutrients and certain toxins as we break things down in the digestive process. We then deliver those nutrients and toxins through the hepatic portal vein to our capillary bed in the liver. And then those liver cells are able to actually take up and process and filter out those nutrients and toxins. And the ones that are not filtered out will return to the hepatic vein and then return to the heart to be circulated through pulmonary circulation via the inferior vena cava. And you can see what this might look like if we were looking at some gross anatomy here. So that is going to conclude part 1 of our Module 4 lecture, and in part 2 we're going to be picking up with physiology, maybe or primarily looking at pulse and blood pressure. And so we'll see you at the next one.